So imagine for a second that you could just freeze time, like completely stop it.
Okay,
And you're looking at the exact microscopic gap in the human brain where a biological electrical signal actually turns into a thought or, a mood or even a profound psychiatric vulnerability.
right, the synapse.
Exactly. But picture it like a vast, dimly lit chamber. It's cavernous, almost like the inside of this colossal cathedral, but instead of stone columns, it's filled with these towering, complex structures.
Like organic relay stations.
Yeah, exactly. And there are these electric blues and sharp, blinding white lights pulsing between the towers. These are the signals just racing along incredibly intricate
And
pathways.
they don't just travel in a straight line, do they?
No, they don't. They reach the edge of a pathway, and there is a physical chasm, a literal gap. And when the signal hits that gap, it doesn't just jump across as a simple white spark.
It changes.
It fractures.
Yeah.
It bursts into these incredibly soft, luminescent purples and greens, and every single color represents a different possible outcome.
Like a different chemical messenger, or a different interpretation by the receiving tower.
Right. Some of these branching pathways flare up brilliantly, while others just instantly fade to black.
It's a really powerful visualization. Because that gap you're describing, that's the microscopic space between neurons where communication actually happens.
Where the magic happens, yeah.
It's where the hardwired electrical impulse of the brain has to, translate itself into a chemical language just to cross the void.
And to guide us through this today on this deep dive, we're going to imagine a detective standing right at the edge of one of these synaptic gaps. Let's call him Medlock Holmes.
I like that. Medlock Holmes.
He's standing there watching this light fracture into purples and greens. He holds a small device, triggers it, and the entire scene just slows down to a crawl.
So the fracturing light freezes in midair.
Exactly. He isn't looking at a single broken wire. He's looking at the precise point where a biological mechanism becomes a human interpretation. He's studying the invisible machinery of thought.
Where every single signal carries, infinite possibilities. And what Medlock Holmes is doing in that chamber, that is the ultimate goal of modern psychiatric genetics.
That's our mission today, right? To step into that chamber with him.
Yeah, we want to trace the path of information. He wants to know exactly where, how, and why a signal might deviate from its expected trajectory.
The deviations that lead to what we clinically categorize as psychiatric disorders.
Precisely. And to really understand this, we have to completely abandon an old framework. If you're familiar with basic biology, you probably know the Mendel's P's model.
Oh, yeah. High school biology.
Right. The deterministic idea that a single specific mutation causes a specific disease. You have the dominant or recessive allele, and boom, you get the trait. It's binary.
But that doesn't work for the brain.
Not at all. When it comes to the architecture of the human brain, that deterministic model is practically useless. We're moving into a world of probabilistic associations.
So it's not just finding a typo on a single page of DNA.
No, we are tracing risk through the entire genome. We're looking for incredibly complex, subtle patterns distributed across millions of genetic variations. It's called polygenicity.
Polygenicity. Which makes the detective work infinitely harder, I imagine.
Oh, it's a nightmare.
Yeah.
But before a geneticist even picks up a pipette to look for these complex patterns,
Right.
before they even look at a strand of DNA, they have to mathematically prove that a mental health disorder actually runs in families.
Because if a condition is entirely driven by the environment, there's no genetic map to find in the first place.
Exactly. And this is the realm of genetic epidemiology. The foundational step is establishing familial aggregation.
Which means proving it runs in the family.
Right. Epidemiologists use a metric called the familial relative risk, which is denoted by the Greek letter lambda.
Okay, lambda, how does that work?
It's a ratio. You basically compare the rate of a disease among the relatives of an affected person to the baseline rate of that disease in the general population.
So if lambda is significantly greater than 1, you have a signal that there might be a genetic cause.
Might be. But as you know, family clustering alone is completely insufficient proof.
Right, because families share DNA, but they also share diets, zip codes, trauma, behavioral environments.
Exactly. If an entire family works in a coal mine,
Yeah.
and they all develop respiratory issues,
You don't go looking for a bad lung gene.
no. You look at the coal dust. You have a shared high-risk environment.
So the statistical challenge is isolating the biological blueprint from the environmental noise. How do they actually do that?
Which brings us to the gold standard, twin studies.
Oh, of course.
Because monozygotic or identical twins share essentially 100% of their genetic material.
Hmm.
And dizygotic, or fraternal, twins share about 50%.
So we can calculate heritability by comparing their concordance rates.
Right. If a disorder is purely genetic, monozygotic twins should have 100% concordance rate. If one has it, the other has it.
And the numbers the data provide on this are just staggering. Let's look at autism spectrum disorder.
The concordance rate there is massive.
Yeah, for identical twins, it's estimated between 80 and 92%. That proves an incredibly strong genetic foundation.
But the delta between identical and fraternal twins is where the math gets really interesting.
Right, because for fraternal twins, that concordance rate drops all the way down to between 1 and 10%.
It's a huge drop.
It's not just a drop, it's a statistical cliff.
It is a cliff.
Yeah.
And it tells us something profound about the genetic architecture. A drop from 90% down to, say, 5% isn't just a quantitative shift.
What does it prove?
It proves that the genetics are not simply additive. If a disease were caused by just a few genes adding their independent risks together, you'd expect the fraternal twins to have roughly half the concordance rate.
Because they share half the DNA, so it should be like 40 or 45%.
Exactly. The fact that it drops to near zero proves we're dealing with epistasis.
Epistasis, that's a gene-to-gene interaction, right?
Yes. Where the effect of one gene is entirely dependent on the presence of another gene somewhere else in the genome.
So it's like a biological combination lock.
That's a great way to put it.
If the combination is 8, 12, 45, 7, an identical twin has the exact same combination, the lock opens, the disorder manifests.
But a fraternal twin only inherits half of those specific numbers.
And having half a combination lock sequence doesn't open the lock halfway, just doesn't open at all.
Exactly. It's an all-or-nothing threshold based on incredibly complex combinations. And we see highly complex heritability profiles in other areas too.
Like bipolar disorder.
Yeah, bipolar has an estimated heritability of 60 to 80%. Schizophrenia sits around 70%.
And major depression is lower, I think, sitting at about 30 to 40%.
Correct.
But here is where I have to push back a little on the history of this science.
Okay, go for it.
If we've known from these twin studies for over half a century that conditions like schizophrenia and bipolar are 70 to 80% heritable, if we knew the combination lock existed, why did it take decades to actually find the genetic numbers?
It's a very fair critique of the field's early optimism. The delay was caused by a massive underestimation of what we call etiological complexity.
Etiological complexity, meaning what exactly?
Well, we knew the traits were heritable, but we assumed they would operate under simpler rules. Psychiatric diseases are governed by several confounding biological realities.
Like what?
First, we have incomplete penetrance. A person can carry the exact disease-related genotype, they have the exact combination lock sequence, but they never actually express the phenotype.
Wait, why? If they have the exact genetic code for the disease, why doesn't the disease trigger the lock never opens?
Because of cellular and environmental buffering. Perhaps they have protective epigenetic markers, or their environment lacked the specific physiological stressor required to activate those risk genes.
Oh, wow. So having the genes doesn't guarantee the disease.
Not at all. Second, you have the inverse problem, phenocopies.
Phenocopies.
These are individuals who develop the exact clinical symptoms of the disorder, but their condition is caused entirely by environmental trauma or secondary medical issues.
Not the genetic risk factors being studied.
Right. When you include them in a genetic study, they act as massive statistical noise.
They dilute the data. You're looking for a genetic signal, but a chunk of your subjects don't actually have the genetic version of the disease.
Exactly. Third, and perhaps the most frustrating for early gene hunters, is locus heterogeneity.
Okay, what does that mean?
This means that entirely different genetic mutations can cause the exact same clinical disease in different family lines.
So family A has schizophrenia from one mutation, and family B has it from a completely different one.
Yes. Imagine family A's schizophrenia is driven by a mutation on chromosome 6, but family B's is driven by a mutation on chromosome 1.
And if a researcher combines the DNA from family A and family B to boost their sample size,
the statistical signals completely cancel each other out.
it's like investigating a series of identical plane crashes. In one crash, the software failed. In another crash, the turbine blade snapped off.
Right.
If you try to find the one single flaw common to all the crashed planes, you will find absolutely nothing because the mechanical failures are totally different, even though the result looks exactly the same.
That is precisely what happened in the 1980s and 90s. And finally, The ultimate hurdle is polygenic inheritance.
Which we touched on earlier.
Right. For the vast majority of these disorders, risk doesn't come from a single broken turbine blade. It comes from thousands of tiny microscopic stress fractures scattered across the entire plane.
And individually, each genetic variant, each microscopic fracture does absolutely nothing.
Exactly. It's only their cumulative weight that pushes the biological system toward a threshold of disease.
Which means we needed a completely different way to look at the DNA. I mean, you can't find microscopic stress fractures across millions of parts using a magnifying glass.
No, you need entirely new mathematical tools. You have to map the invisible.
So what tools did they come up with?
This led to the development of the two fundamental strategies in the Gene Hunter's Toolkit: linkage analysis and association studies.
And it's vital to understand the mechanical difference between these two, right? Because they are designed to hunt for entirely different types of genetic architecture.
Exactly. Let's begin with linkage analysis. This method requires specific multi-generational family pedigrees.
So looking at huge family trees.
Yes. It's a tool built exclusively to find rare genetic variants that have a massive, highly penetrant effect on a disease.
When I look at the math behind linkage analysis, it relies entirely on the biological mechanics of meiosis, right?
Yes, the way cells divide to create eggs and sperm.
The crossing over process.
Right. Biologically, linkage leverages the phenomenon of chromosomal recombination. During meiosis, the homology chromosomes line up and physically swap chunks of DNA to ensure genetic diversity.
However, genes that are physically located right next to each other on the chromosome are highly unlikely to be separated during this swapping process.
Exactly. They travel together, they are linked.
So if you have a family where a very specific rare disease is being passed down, researchers look for genetic markers that are consistently inherited alongside the disease within that specific family tree.
Precisely. If the marker and the disease are always passed down together from grandmother to father to daughter, they are said to be inherited identical by descent, or IBD.
And the closer the marker is to the actual disease-causing mutation, the less likely they are to be split up.
Right. Geneticists measure this distance using the recombination fraction, denoted by theta.
Let's clarify theta because the math is important here. If theta is 50%, what does that actually mean?
If theta is 50%, or 0.5, it means the genes are completely unlinked. They are segregating randomly, basically a coin flip.
But if theta is very low, say 0.01.
It means they're tightly linked and almost never cross over separately. To prove this isn't just random chance, geneticists calculate a LOD score.
Which stands for the logarithm of the odds.
Yes. It compares the likelihood that two loci are linked versus the likelihood that they are unlinked. A LOD score of 3 means the odds are 10 to the power of 3, or 1,000 to 1, that the marker and the disease gene are physically linked.
1,000 to 1 odds sounds fantastic, but the source text makes it painfully clear that for psychiatric disorders, linkage analysis largely ended in tears.
It did. It chased statistical ghosts.
It assumed psychiatric genetics were driven by a few heavy-hitting mutations in specific families, and when that turned out to be false, the field had to pivot.
They pivoted to the second, vastly more complex tool, association studies, specifically genome-wide association studies, or GWAS.
Okay, so if linkage uses families to find rare variants of massive effect, what does GWAS do?
GWAS uses massive populations of completely unrelated people to find common variants of incredibly small effect.
So you're no longer looking at how DNA is passed down from a grandfather.
No, you're looking at the entire human population as one giant, ancient family tree.
And the biological mechanism that makes GWs possible is completely different than the meiotic crossing over in a single family. GWAS relies on something called linkage disequilibrium, or LD.
Yes, LD is the cornerstone of population genetics. Over tens of thousands of generations, human chromosomes have been chopped up and recombined by meiosis so many millions of times that the original ancestral chromosomes are completely shattered.
But very tiny microscopic blocks of DNA have survived this chopping process intact.
Exactly. These tiny surviving blocks are called haplotypes.
So if a specific genetic variant and allele within one of these tiny haplotype blocks occurs in people with the disease more often than you'd expect by pure random chance across a population of millions, you have an association.
Right. They are in linkage disequilibrium. You found a fingerprint of risk.
But the statistical danger here is monumental. You mentioned a LOD score of 3 for linkage.
Yeah.
But in GWS, researchers aren't testing one marker. They are using microarrays to test millions of tiny genetic variants called single nucleotide polymorphisms, or SNPs, across the entire genome, all at exactly the same time.
And anyone who understands basic statistics knows the multiple testing problem.
Right. If you run 5 million independent statistical tests, pure random chance guarantees that you will find tens of thousands of significant associations just by a fluke of the numbers.
Which is why the field had to adopt a brutal statistical correction, usually the Bonferroni correction.
How brutal are we talking?
To eliminate these false positives, the threshold for genome-wide significance in a GWAS is a p-value of 10 to the negative 7th power.
10 to the negative 7th. That is a staggering threshold.
It requires massive sample sizes, tens or hundreds of thousands of people, just to generate enough statistical power to prove that a tiny genetic variant is actually associated with a trait and not just a mathematical hallucination.
It really is a marvel of computational biology, but here is the critical flaw that haunted the field for years.
The phenotype problem.
Right. You can have the most powerful supercomputers in the world. You can calculate p-values to the negative 10th power, but none of that math matters if the actual data you feed into the machine is conceptually flawed.
And that brings us to the most frustrating roadblock in psychiatric genetics, the phenotype problem.
Wait, so we have the technology to read the DNA perfectly, but the problem is we don't actually know how to define the disease we are trying to cure.
Exactly. A phenotype is the observable physical trait or clinical presentation we are trying to map. In a field like cardiology, the phenotype is highly objective.
You look at an angiogram and you see a physical plaque blocking an artery. It's biologically undeniable.
But in psychiatry, our phenotypes for the last century have been categorical diagnoses entirely based on the DSM, the Diagnostic and Statistical Manual of Mental Disorders.
And our deep dive sources offer a brilliant, highly critical breakdown of why the DSM is a nightmare for geneticists.
It operates on a menu-based system of symptoms.
Right. Let's take major depressive disorder. The manual states you need to meet 5 out of 9 specific symptoms to receive the diagnosis.
Right.
So picture 2 different patients. Patient A walks into the clinic presenting with depressed mood, severe weight loss, severe insomnia, psychomotor agitation, and crippling fatigue. That's 5 symptoms.
They get the diagnosis of major depression.
Now, patient B walks in presenting with a complete loss of interest in hobbies, massive weight gain, hypersomnia, sleeping 14 hours a day, feelings of utter worthlessness, and suicidal ideation. That is also 5 symptoms.
And they receive the exact same categorical diagnosis of major depression.
But look at the clinical reality. Those 2 patients have absolutely 0 overlapping symptoms. Patient A is starving and awake. Patient B is overeating and unconscious.
Right. So if their biological symptoms don't overlap in the slightest, why on earth would a geneticist expect their underlying DNA mutations to overlap?
It is absolute madness to put them in the same genetic cohort.
It's like we've been classifying vehicles purely by their exterior paint color. We put all the red vehicles in one study group and all the blue vehicles in another.
And then we run a billion-dollar genetic analysis, open the hoods of the red group, and are completely shocked to discover that one is a Ferrari, one is a diesel tractor, and one is a bicycle.
Exactly. The symptom profile, the paint color, tells us absolutely nothing about the mechanics of the engine.
That is one of the most accurate analogies for the history of psychiatric nosology. By grouping biologically disparate mechanisms under single clinical labels, we mathematically diluted our own genetic signals.
We created our own statistical noise. When researchers finally accepted this limitation, it sparked a paradigm shift toward mapping continuous phenotypes.
Specifically what are known as endophenotypes.
Yes, so if the DSM diagnosis is the paint color, the endophenotype is classifying the vehicles by the actual mechanics of their engines.
Okay, so what actually is an endophenotype?
It's a measurable, continuous underlying biological trait that is conceptually closer to the genes than the broad clinical diagnosis.
Because these traits have a much simpler genetic architecture.
Right. So instead of trying to find the genes for a massive, blurry concept like schizophrenia, what if we map the genes responsible for hippocampal volume, as measured on an MRI?
Or map the specific electrical oscillations of the brain using an EEG.
Or map highly specific neurocognitive deficits, like a failure in working memory, because the genes themselves have no idea what the DSM is.
Right. Genes don't code for schizophrenia. They code for specific proteins that build a specific calcium channel in the hippocampus.
Yes. And to be a valid endophenotype, a trait must meet strict criteria. It must be heritable. It must co-segregate with the illness in families,
And crucially, it must be state independent.
meaning the biological marker must be present in the individual, even when they are not actively experiencing a psychiatric episode. It's a baseline structural or functional vulnerability.
And what's amazing about shifting to endophenotypes is that it finally allowed scientists to use animal models in a way that actually produced valid data.
You can't put a mouse on a couch and ask if it's feeling existential dread.
No. And you can't diagnose a fruit fly with bipolar disorder using the DSM, but you absolutely can measure their biological endophenotypes.
The text provides an incredible historical example of this regarding circadian rhythms.
Walk us through that because it's fascinating.
Well, severe alterations in sleep and daily activity patterns are profound, core features of mood disorders, particularly the manic phase of bipolar disorder.
Okay.
In the 1970s, geneticists who were mapping the genome of Drosophila fruit flies discovered a specific gene that controlled the fly's biological clock. They named it the period gene.
And decades later, scientists found the mammalian ortholog of this gene in mice.
Right, literally named the clock gene.
And this is where the biology is just crazy. When scientists genetically engineered mutant mice with a targeted disruption in this clock gene, the mice didn't just sleep at weird times.
What did they do?
They displayed a behavioral profile that was shockingly similar to a human manic episode. They were hyperactive, they showed decreased need for sleep, and they engaged in risk-taking behaviors.
They mirrored the exact endophenotype of mania.
But the truly staggering confirmation of this model came when they tested pharmacological interventions on these mutant mice. How did they treat the hyperactive clock-mutated mice?
They administered lithium.
The exact same elemental mood stabilizer that has been the gold standard treatment for human bipolar disorder for decades.
Exactly. And the lithium effectively modified the manic-like behavior in the mice and stabilized their circadian activity.
This is the power of the endophenotype approach.
By focusing on a specific, measurable biological mechanism, circadian rhythmicity researchers traced a direct line from a genetic mutation to an observable cellular deficit to a complex behavior and ultimately to a proven pharmacological treatment.
It's a stunningly elegant pathway. But before we try to apply this highly complex logic to the blurry boundaries of all psychiatric disorders, we should look at a field where both linkage analysis and GW worked exactly as intended.
Right, the biological template for success. Alzheimer's disease.
Let's talk about Alzheimer's. Why is it the perfect case study?
It illustrates how different genetic tools uncover different layers of a disease. Unlike psychiatric disorders, Alzheimer's is a neurodegenerative dementia with a very clear physical footprint in the brain.
If you do an autopsy, you can physically see the biological crime scene.
Exactly. You see extracellular senile plaques composed of beta-amyloid fibrils and intracellular neurofibrillary tangles made of hyperphosphorylated tau protein.
Right. The endophenotype is literally visible under a microscope. And epidemiologists noticed two very distinct patterns of Alzheimer's inheritance in the general population.
The vast majority of people developed it sporadically in old age.
But there was a very rare early-onset familial form that struck people in their 40s and 50s. And this early-onset form accounted for only about 5% of all cases. But it seemed to be passed down in a strict Mendelian autosomal dominant pattern.
Which, as we discussed, is the absolute perfect scenario for linkage analysis. You have dense, multi-generational families with a highly penetrant trait.
So geneticists tracked the disease through these specific pedigrees.
And the results were a massive breakthrough. By the early 90s, they mapped a mutation on chromosome 21 to the APP gene.
APP stands for amyloid precursor protein.
Right. Shortly after, linkage in other affected families found mutations on chromosome 14 in a gene called presenilin 1 and on chromosome 1 in presenilin 2.
Okay, this is where I want to dive into the physical mechanics because it's so satisfying when the genetics perfectly explain the biochemistry.
Let's do it.
What do these three totally different genes on three different chromosomes actually have in common?
They are all critical components of the exact same biochemical assembly line. The APP gene produces a long precursor protein that sits across the cell membrane of the neuron.
Okay, the raw material.
To function normally, this long protein has to be cut into smaller pieces by enzymes. The presenilin genes, PS1 and PS2, are the catalytic core of a massive enzyme complex called gamma secretase.
So gamma secretase acts as the biochemical scissors that snip the APP protein.
Correct. In a healthy brain, gamma secretase cuts APP to produce a soluble fragment called A-beta 40, which is easily cleared away.
But when a patient has a mutation in either the APP gene or the presenilin genes,
It physically alters how the scissors align with the protein. Instead of cutting at position 40, it cuts at position 42, producing a beta 42.
and that's bad.
Very bad. This slightly longer fragment is highly hydrophobic and sticky. It clumps together, forming the toxic insoluble plaques that physically crush and destroy the surrounding neurons.
It's incredible. Linkage analysis completely cracked the core molecular biology of the disease. It proved the amyloid hypothesis.
But that only explained 5% of the patients.
Right. What about the other 95%, the people who get late-onset Alzheimer's in their 70s and 80s?
When researchers ran linkage analysis on late-onset families, they found absolutely nothing. The LOD scores were flat.
So those rare, heavy-hitting presenilin mutations simply did not exist in the broader population.
Exactly. The genetic architecture was fundamentally different. It was polygenic. So the field transitioned to association studies to look for common variants.
And in 1993, using early association techniques, they discovered what remains the strongest genetic risk factor for any common human disease.
The ApoE4 allele on chromosome 19.
And ApoE isn't a pair of scissors. It has a completely different job. It's involved in lipid metabolism, right? Moving cholesterol around the brain.
Yes, apolipoprotein E is a lipid transporter. But crucially, in the brain, it is also secreted by astrocytes and microglia, the brain's immune and support cells, to bind to beta amyloid and help clear it out of the extracellular space.
And there are 3 common variants of this gene in humans, E2, E3, and E4.
And the E4 variant is physically less efficient at binding and clearing that toxic amyloid.
So if you inherit one copy of the inefficient E4 variant from a parent, your statistical risk of developing late-onset Alzheimer's increases about 4-fold.
If you inherit 2 copies, one from each parent, your risk skyrockets 10 to 15-fold.
Wow. But it isn't deterministic like the presenilin mutations, right? You can have 2 E4 alleles and still never get the disease.
Exactly. It just fundamentally shifts your biological probability. And as GWAS microarray technology exploded in the 2010s, we didn't just stop at ApoE.
Massive GWAS consortia have now identified over 40 distinct genetic loci associated with late-onset Alzheimer's.
And what blows my mind is that these 40 new genes aren't just more amyloid scissors. They are pointing to completely different systems in the body.
The sources highlight genes like CD33 and CR1. What are those doing in the brain?
CD33 and CR1 are intimately involved in the innate immune system and inflammatory cascades.
The immune system.
Yeah, this was a massive paradigm shift. It suggested that the root cause of late-onset Alzheimer's isn't just the overproduction of amyloid, but a failure of the brain's immune system, specifically the microglia, to properly manage the clearance of cellular debris.
The immune cells might become chronically activated, and the resulting neuroinflammation might actually be the primary driver of neuronal death.
Precisely, and we're going to see the immune system pop up again in a very surprising place later.
But perhaps the most exciting finding in the Alzheimer's genetic story isn't about mapping risk or inflammation at all. It's about mapping resilience.
Right. Because you can perform an autopsy on a 90-year-old, look at their brain, and find it absolutely packed with toxic amyloid plaques.
Yet when that person was alive, they exhibited 0 cognitive decline. Their memory was perfectly intact.
How does the biological hardware survive that?
Researchers recently discovered a fascinating, highly protective genetic variant known as the A673T mutation.
It's a single missense mutation located in the APP gene, exactly adjacent to where another enzyme, beta secretase, makes its initial cut before gamma secretase finishes the job.
So it's a typo right at the cutting site.
Yes, and this specific typo physically alters the shape of the cleavage site just enough to make it significantly harder for the beta secretase enzyme to bind and cut the protein.
And as a result of this tiny structural hindrance, the overall lifetime production of the toxic A-beta 42 fragment is literally reduced by about 40 to 50%.
Individuals carrying this extremely rare mutation are profoundly protected against Alzheimer's disease and cognitive decline, even if they live well past 100 years old.
Which fundamentally changes the objective of the science. We aren't just looking for broken genes so we can fix them. We are searching the human genome for naturally occurring instances of biological resilience.
If we understand the exact mechanism of that protective mutation, pharmaceutical companies can design a drug that physically mimics that exact structural hindrance at the beta secretase cleavage site.
Which is exactly what the field of BACE inhibitors attempted to do, though translating that to clinical success remains a profound challenge.
But Alzheimer's provided a relatively clear roadmap: tangible plaques, specific enzymes, measurable clearance rates.
However, As we shift our focus to neurodevelopmental conditions, that clean roadmap just vanishes.
Completely. We have to look at what happens when the genetic variations don't cause a degenerative disease in old age, but fundamentally alter how the microscopic architecture of the brain is built from infancy.
We are talking about autism spectrum disorders.
Autism introduces a level of biological complexity that makes Alzheimer's look like simple arithmetic.
Autism is defined as a neurodevelopmental disorder characterized by impaired social communication, altered sensory processing, and restricted repetitive behaviors. And genetically, it presents a massive paradox.
Right. On one hand, as we established with the twin studies, identical twins have up to a 92% concordance rate. It's incredibly heritable.
But traditional family pedigree studies show a massive drop-off in risk between first-degree relatives and second-degree relatives, which confirms extreme epistasis.
So how do you map a condition that is highly genetic but refuses to follow simple inheritance rules?
Researchers started by looking at the largest, most disruptive genetic typos possible, copy number variations, or CNVs.
I want to make sure we clearly define a CNV, because it is very different from the single-letter typos, the SNPs we talked about in GWAS.
Yes. A single nucleotide polymorphism is a change in one single letter of the DNA code. An A becomes a G.
But a copy number variation is a massive structural aberration. Entire chunks of a chromosome, spanning thousands or even millions of base pairs and containing dozens of entire genes, are either physically deleted or duplicated.
Biologically, this usually happens due to unequal crossing over during meiosis. Imagine someone accidentally highlighting 3 entire pages in a manual and hitting delete, or hitting copy-paste twice.
The structural integrity of the genome in that region is profoundly altered.
And the source text notes that these large-scale de novo CNVs, meaning they are new mutations not inherited from the parents, account for about 5 to 8% of what we call syndromic autism cases.
But just finding a missing chunk of a chromosome doesn't tell you the mechanism. You have to know what those missing genes were supposed to be doing.
And to figure that out, scientists look closely at rare, specific syndromes where autism is a core clinical feature.
Exactly. They looked at monogenic syndromes like Fragile X, tuberous sclerosis, and Rett syndrome. These conditions have simpler, often single-gene inheritance patterns, which makes them easier to map.
Fragile X, for instance, is the most common inherited cause of autism.
It is caused by a massive expansion of a repeating DNA sequence in the FMR1 gene on the X chromosome. This expansion eventually silences the gene, shutting off production of the FMRP protein.
And what does the FMRP protein actually do in a healthy brain?
It acts as a translational suppressor at the synapse. It binds to messenger RNA and physically transports it from the nucleus down the long axon to the synaptic terminal, holding it in a paused state until the synapse receives an electrical signal.
Only then does it allow the RNA to be translated into new proteins to strengthen the synaptic connection.
Without FMRP, protein synthesis at the synapse is unregulated and chaotic.
Wow. What about the other syndromes? Do they also impact the synapse?
Yes, tuberous sclerosis involves mutations in the TSC1 or TSC2 genes. These genes normally suppress a massive cellular growth pathway called MTOR. When they are mutated, the neurons grow abnormally, disrupting the physical branching structure of the dendrites.
And Rett syndrome.
A devastating condition that primarily affects girls is caused by a mutation in the MECP2 gene. MECP2 is an epigenetic reader. It binds to methylated DNA and controls the expression of thousands of other genes required for a neuron to fully mature and form stable synapses.
So when you aggregate all the biological data from all these different rare syndromes, Fragile X, tuberous sclerosis, Rett's, they all converge on the exact same location.
They all point directly to the formation, maintenance, and plasticity of the synapse. Which brings us right back to our opening visual with Medlock Holmes.
Exactly. When Medlock Holmes is watching that signal fracture across the gap, he is looking at the physical manifestation of these genetic instructions.
The genetics of autism are revealing that the core biological vulnerability lies in the physical and chemical scaffolding that builds and operates that synaptic gap.
Let's talk about the specific proteins that make up that scaffolding, because the genes highlighted in the text are fascinating. It specifically mentions the neuroligin family of genes, or NLGN.
Neurologens are essentially the glue of the mind. They are cell adhesion molecules located on the postsynaptic membrane, the receiving side of the gap.
And they reach across the synaptic cleft and physically bind to partner proteins called neurexins, specifically NRXN1, which are anchored on the presynaptic membrane, the transmitting side.
So they are literally physical cables tying the two neurons together across the void to keep the connection stable during rapid-fire communication.
Yes. And when researchers genetically engineered mice with mutations in these neurolineage genes, the mice demonstrated impaired social novelty preference and significantly reduced ultrasonic vocalizations.
Which are the mouse behavioral equivalents of human autistic traits.
And behind the neurologens inside the receiving cell, you have the Schenk gene family, specifically Schenk3.
Schenk3 encodes the master organizing proteins that build the postsynaptic density. Think of it as the concrete foundation of the receiving tower. It anchors the neurotransmitter receptors exactly where they need to be to catch the chemical signal.
If you have a mutation in the Schenk foundation, or the neuroligin glue, or the neurexin cables, the signal that Medlock Holmes is watching does not fracture or transmit correctly.
The timing is off. The chemical interpretation is altered.
Okay, I follow the biology completely. It makes perfect sense. But hold on, I need to push back here.
Sure.
Because everything you just described, the massive missing chromosome chunks, the chaotic protein synthesis, the broken physical scaffolding, it sounds incredibly destructive. It sounds like a broken machine.
It does.
But autism is a massive spectrum. How does a massive destructive deletion explain the highly functional, often brilliant individuals with Asperger's or high-functioning autism? You can't have a totally broken synaptic scaffold and simultaneously have an IQ of 140 and a profound capacity for systems thinking.
That is the pivotal question. And it is exactly why autism genetics is so revolutionary. The architecture we just described, the CNVs and severe syndromic mutations, explains the rare, highly penetrant forms of autism, which are very often comorbid with severe intellectual disability and epilepsy.
But what about the vast majority of the spectrum, the familial forms?
To answer that, we have to look at a staggering insight that emerged from a massive 50,000-person GWAS study.
This is the finding that completely rewired my understanding of neurodiversity. Walk us through exactly what that GWAS revealed.
The GWAS Consortium took their massive data set of common polygenic variants associated with autism and mathematically cross-referenced it with an independent GWAS data set that had mapped the genetics of educational attainment and general cognitive function.
And what they found split the biological architecture of the spectrum in half.
Exactly. Non-familial syndromic autism, the cases driven by those large, destructive de novo CNV mutations, are strongly linked to cognitive impairment. It is fundamentally a disruption of normal development.
But the familial cases of ASD, the polygenic risk derived from thousands of tiny common variants passed down through generations, showed a massive positive genetic correlation with higher educational achievement, elevated intelligence, and enhanced cognitive performance.
Let that completely sink in for a second. The exact same common genetic variants that increase a family's polygenic risk for higher functioning autism are biologically and mathematically intertwined with the genetic factors that increase intelligence.
The biology of the autistic mind in its polygenic form is functionally linked to enhanced cognitive capacity. It implies that certain neurodivergent traits aren't a defect or a broken machine at all, but are the natural result of genetic pathways optimizing for highly specific types of deep, systemic cognitive processing.
It forces a profound reevaluation of how we define pathology versus natural human biological variation. It suggests that the spectrum actually encompasses two entirely different genetic realities that happen to produce superficially similar behavioral traits.
It is a phenomenal realization. But as profound as that split is, autism still maintains relatively clear diagnostic boundaries in the clinic. A psychiatrist can usually tell the difference between autism and other major psychiatric conditions.
However, as we wade deeper into the genetic map, we enter what I call the messy middle. We are looking at bipolar disorder and schizophrenia. And in this realm, the clinical boundaries we have trusted for a century completely dissolve.
The history of mapping bipolar and schizophrenia is arguably the most dramatic and brutal chapter in psychiatric genetics. It is a story of immense hubris, intense rivalries, and crushing false positives. Let's start with bipolar disorder.
The text highlights a specific infamous moment in 1987.
Right. In 1987, two different research teams published papers in prestigious journals announcing massive, definitive breakthroughs. One team had studied multi-generational families in Israel where bipolar disorder seemed to be inherited perfectly alongside color blindness.
Since color blindness is mapped to the X chromosome, They declared they had found the locus for bipolar disorder on the X chromosome.
At the exact same time, a competing team published a study on a massive old-order Amish pedigree in Pennsylvania. Bipolar disorder was rampant in this specific family tree, and using linkage analysis, they mapped the disease locus definitively to the short arm of chromosome 11.
And they didn't just guess, they had the mathematical proof. The LOD scores for these studies were incredibly high, ranging between 4 and 9.
Remember, a LOD score of 3 is 1,000 to 1 odds. A LOD score of 9 is astronomically certain. It looked like definitive Nobel Prize-winning proof. They had found the bipolar genes.
But it was a complete illusion. It was a statistical mirage. When independent laboratories attempted to replicate the chromosome 11 finding in different populations, they found absolutely nothing.
But even worse, when the original researchers went back to that exact same Amish family a few years later, updated the clinical diagnoses of younger family members who had newly developed the disorder, and reran the analysis with better DNA markers, the original signal completely vanished. The linkage evaporated into thin air.
I need you to explain the math here, because how does an astronomical LOD score of 9 just evaporate? The math is supposed to be objective.
The math was objective, but the biological assumptions fed into the equation were entirely wrong. The researchers were looking at a highly complex polygenic trait bipolar disorder, but they forced it into a simple, single-gene Mendelian statistical model.
When you have small family sample sizes and you apply overly optimistic mathematical parameters regarding penetrance and disease frequency, random variations in how the genes segregate can artificially inflate the LOD score to absurd levels.
It was a statistical hallucination caused by underpowered studies and the assumption of a single dominant gene.
It was a brutal lesson in etiological complexity. It took decades for the field to recover its credibility. They realized they had to abandon linkage and build massive international consortia to run properly powered GWAs.
They had to pool data from the Wellcome Trust Case Control Consortium in the UK, the STEP program in the US, and University College London. They needed the DNA of over 10,000 people just to punch through the statistical noise and find the very first real, confirmed genetic hit for bipolar disorder.
And that first confirmed hit was the ANK3 gene. ANK3 encodes a massive scaffolding protein called ankyrin G.
Let's do the biology. What does ankyrin G do in the brain?
Think about the long axon of a neuron acting like an electrical wire. To fire an electrical impulse efficiently, the neuron has to cluster thousands of voltage-gated sodium channels at very specific, uninsulated gaps along the wire, known as the nodes of Ranvier.
Ankyrin G is the master organizer. It physically anchors all those sodium channels in place.
If ANK3 is slightly mutated, the clustering of the channels is altered, and the electrical firing threshold of the entire neuron becomes unstable. It might fire too easily or misfire.
Which perfectly aligns with the clinical reality of bipolar disorder, a disease of neurological instability and overexcitability. And just like the clock gene, when researchers knocked out ANK3 in the forebrains of mice, the mice developed a manic-like phenotype that was subsequently rescued by administering lithium.
The biology aligns perfectly. They also found a second major hit in a gene called CACNA1C, which is a calcium channel gene. We're going to come back to that one because it is the ultimate multitasker. But if bipolar was hard to map, the story of schizophrenia is even more chaotic.
Schizophrenia mapping went through a very similar phase of false hope. During the early days of association studies, researchers found a whole host of what they called candidate genes based on small, localized family studies.
You might recognize the ominous names: Dispendin, Neuregulin, and DISC1, which literally stands for Disrupted in Schizophrenia 1.
And neurobiology laboratories spent millions of dollars and years of labor building genetically engineered mutant mice for every single one of these candidate genes. The mice showed fascinating behavioral deficits. The neurodevelopmental biology seemed incredibly compelling.
But the human genetics did not hold up. When they scaled up the studies to larger human populations to confirm the associations, the replication was incredibly messy. The statistical evidence for DIC1 and Dispendin was equivocal at best.
The field realized they were vastly underpowered, so they decided to throw the absolute kitchen sink at the problem.
In 2014, the Schizophrenia Working Group of the Psychiatric Genomics Consortium assembled a sample size that was previously unfathomable. They pooled DNA from 37,000 patients with schizophrenia and 113,000 healthy controls. 150,000 people in a single study.
And with that massive statistical gravity, the signal finally punched through the noise. They identified 108 distinct genetic locations associated with schizophrenia.
It was a watershed moment. But the most prominent, undeniable hit among those 108 loci was a massive signal sitting right on chromosome 6. Specifically, it mapped directly to a region called the major histocompatibility complex, or the MHC.
Okay, hold on. This is where I have to stop and demand an explanation. Anyone who has taken Biology 101 knows what the MHC is. It is the absolute command center for the human immune system.
It is how your white blood cells recognize a foreign virus, or why your body rejects a transplanted kidney.
What on earth is an immune system command center doing as the #1 genetic signal for a psychiatric disorder defined by auditory hallucinations and delusions? How does a white blood cell alter a thought?
It is a profound finding, and it completely revolutionized our understanding of neurobiology. We have known from epidemiological studies for a long time that severe maternal viral infection during the second trimester of pregnancy is a major environmental risk factor for schizophrenia in the child.
So one hypothesis was that specific MHC genetic configurations lead to an anomalous hyperactive immune response in utero, which alters the fetal brain development.
But the biology goes even deeper than that, right? It's not just about fighting viruses.
Exactly. Researchers later discovered that the neurons themselves and the brain's resident immune cells, the microglia, actively use MHC molecules for an entirely different purpose: synaptic pruning.
During late adolescence, which is precisely when schizophrenia typically onsets, the brain undergoes a massive remodeling phase. The microglia literally eat and prune away weak or unnecessary synapses to streamline the brain's efficiency.
So the immune cells are acting like gardeners, trimming the synaptic hedges.
Yes. And a specific gene within the MHC region called complement component 4, or C4, tags the synapses that need to be destroyed. The GWAS data suggests that certain genetic variations cause an overexpression of C4 in the brain.
The microglia become overly aggressive. They prune too many synapses, physically stripping the cortical networks of their connectivity.
This excessive pruning structurally degrades the brain's ability to maintain coherent reality testing, leading to the psychotic symptoms of schizophrenia.
It is a stunning convergence of immunology and psychiatry. But even with 108 confirmed genetic locations, and even knowing about the C4 pruning mechanism, there's a glaring mathematical paradox in the schizophrenia data.
And this brings us to my favorite concept in the entire text, the missing heritability problem.
It is a massive, highly debated paradox in modern genetics. Let's look at the math. The twin studies from the 1960s unequivocally proved that schizophrenia is about 70% heritable. The combination lock exists.
But if you take all 108 common genetic loci found in that massive 150,000-person GWAS study, and you mathematically add up all their tiny individual odds ratios, they only account for about 30% of the genetic variance.
Wait, let me get this straight. We know from twins that it is 70% heritable. But after scanning 150,000 genomes, we can only find the specific DNA typos for 30% of it. Where is the other 40%? Where is the missing heritability?
That is the multi-billion-dollar question in genomics. The current prevailing hypothesis is that the gap is filled by thousands of ultra-rare genetic variants of medium to large effect.
GWAS microarrays are simply blind to these. GWAS is designed only to look for common variance typos that are shared by thousands of people in the population.
It fundamentally misses the ultra-rare, entirely unique typos that might only exist within a single specific family line.
So to find the missing heritability, we can't just use microarrays that scan for known SNPs. We have to do whole genome sequencing on millions of people. We have to read every single one of the 3 billion letters of DNA in every single patient to find their specific, unique mutations.
Which is technologically staggering, though the cost of sequencing is rapidly dropping. But if you think schizophrenia and bipolar were hard to map, we are now looking at the geneticist's Everest. Because if schizophrenia required 150,000 people to find 108 genes, major depression makes that look like child's play.
Major depression is the most common psychiatric disorder in the world. The lifetime prevalence is estimated at around 8 to 12% of the global population. It is a massive, crushing public health crisis.
But genetically, it is incredibly elusive. The heritability is relatively low. Twin studies put it at only about 30 to 40%.
Which means the vast majority of the variance is driven by the environment. Trauma, chronic stress, socioeconomic conditions, and life events play a massive role in its etiology. The genetic signal is incredibly faint compared to the environmental noise.
And for decades, scientists thought they could bypass the massive population studies because they believed they already knew the biology. They believed the serotonin hypothesis. They assumed depression was caused by a chemical imbalance of serotonin.
So they focused exclusively on candidate gene studies, looking specifically at the genes that build serotonin transporters and serotonin receptors. The text is absolutely brutal in its assessment of this era.
It is a harsh but necessary correction. The text points out that over 1,500 separate investigations looked at roughly 200 of these highly specific candidate genes for depression. Entire careers were built on analyzing the promoter region of the serotonin transporter gene, known as 5-HTTLPR.
But when modern, highly powered statistical methods and massive replication cohorts were finally applied to those 200 genes, almost every single one of them was exposed as a false positive.
It is devastating to think of the wasted time and resources. Aside from perhaps a very minor signal in the DRD2 dopamine receptor, the entire early catalog of depression genetics was essentially a statistical mirage driven by publication bias and underpowered samples.
They realized the problem wasn't necessarily the biology, it was the math. To find the actual genes for a highly prevalent, highly environmental disease with only 30% heritability, you need numbers that defy imagination.
Schizophrenia got its first GWAS hits at around 15,000 people. Major depression didn't get a single confirmed replicable hit until a direct-to-consumer biobank 23andMe ran a study utilizing 75,000 self-reported cases of depression.
75,000 cases just to get a single statistical blip on the radar. And what they found were genetic variants near a gene called NGR1, which is involved in neuronal growth, and OLFM4, which, unsurprisingly, given what we now know, is involved in inflammation and immunity.
But the effect sizes of these variants are infinitesimally small. Each variant gives an odds ratio of roughly 1.04.
An odds ratio of 1.04 means that if you have the variant, your relative risk of developing depression increases by a mere 4%. It is barely a whisper of biological influence.
I picture the math this way. Mapping the genetics of major depression is like standing in a packed, screaming football stadium with 100,000 people, and you are trying to isolate the sound of one specific person whispering in the third row.
You can't do it with a stethoscope. You need 75,000 highly sensitive microphones distributed throughout the entire stadium, and you need supercomputers to filter out the crowd noise of trauma and environment just to isolate that single 4% frequency.
That is exactly right. And for anxiety disorders, the numbers had to be even larger. Anxiety disorders like generalized anxiety and panic disorder have similarly low heritabilities, hovering between 30 and 50%.
Early linkage studies on families with high anxiety found absolutely nothing replicable. To finally crack the genetic code of anxiety, researchers utilized the Million Veterans program, leveraging genomic data from nearly 200,000 individuals.
And what blows my mind about the MVP study is the phenotype they used. They didn't use a massive 3-hour structured psychiatric interview. They used a simple 2-question survey called the GAD2, which just asked people to rate how often they feel anxious or can't stop worrying on a simple sliding scale.
Which is a brilliant, highly pragmatic use of a continuous endophenotype. By turning anxiety into a quantitative sliding scale rather than a binary yes or no clinical diagnosis, they dramatically increased their statistical power.
And in those 200,000 veterans, the signal finally broke through. They found significant associations with genes like PDE4B, which is involved in intracellular signal transduction, and ESR1, which encodes an estrogen receptor, providing a potential biological clue as to why anxiety disorders are statistically more prevalent in women.
But as we pool these hundreds of thousands of genomes together across all these different disorders, something truly radical is happening to the data. As we map the entire architecture of psychiatric vulnerability, the ultimate plot twist is revealing itself.
The genes do not care about the DSM. They do not respect the clinical boundaries we have spent a century building.
This brings us to the profound concept of genetic correlation, or R sub G. Because we now have massive GWAS data for hundreds of thousands of people across multiple different psychiatric disorders, we can use complex mathematics to overlap their genetic risk profiles.
We can ask the computer, how much of the polygenic risk for disease A is shared with disease B? And the results completely dismantled traditional psychiatric nosology. The data reveals a staggering 68% genetic overlap between the common risk variants for schizophrenia and bipolar disorder.
68%. If you look at the symptoms in a textbook, schizophrenia is primarily classified as a psychotic disorder defined by hallucinations, delusions, and cognitive disorganization. Bipolar is primarily classified as a mood disorder defined by the cyclical extremes of mania and severe depression.
But genetically, under the cellular hood, they are sharing nearly 70% of their foundational risk architecture.
And the overlap doesn't stop there. Major depression shares approximately 45% of its polygenic risk with both schizophrenia and bipolar disorder. The biological vulnerabilities are deeply intertwined. Remember that calcium channel gene we mentioned earlier, CAC-NA1C?
Yes, the voltage-gated calcium channel, the ultimate multitasker.
Exactly. Large-scale cross-disorder GWAS has revealed that variations in CAC-NA1C act as a significant genetic risk factor for autism, for bipolar disorder, and D for schizophrenia. This is the biological definition of pleiotropy.
A single genetic variant affects multiple, seemingly distinct phenotypic traits.
So how does one mutated calcium channel cause three different diseases?
It implies that these underlying cellular pathways, like the regulation of calcium signaling during an action potential, do not cause a specific disease. They create a generalized vulnerability in the brain's processing power. They destabilize the network.
The specific clinical manifestation, whether that unstable network develops into schizophrenia or bipolar or autism, is determined by the unique combination of the rest of the person's genetic background and, crucially, their environment.
And the genetic overlap extends far beyond clinical psychiatry. The text points to massive genetic correlations with basic subclinical human personality traits. Major depression has a 0.70 genetic correlation with the psychological personality trait of neuroticism.
It has a highly negative correlation, negative 0.75, with subjective well-being. There are even profound physical overlaps. The polygenic risk for atypical depression, which features weight gain and hypersomnia, is heavily genetically correlated with body mass index, or BMI.
Which proves undeniably that mental health is inexorably linked to physical biology and fundamental personality structure. But the final, most important piece of the puzzle, the factor that bridges the gap between the static DNA sequence and the fluid clinical reality of the patient, is the environment.
With these massive cohorts, we finally have the statistical power to mathematically measure gene-environment interactions, or GXE.
I think of this using a computer analogy. The DNA is the physical hardware. It is the motherboard and the silicon chips. But the environment is the software update. And the text highlights a fascinating study from the UK Biobank looking at the Townsend Deprivation Index.
This is a robust epidemiological measure of socioeconomic status. It looks at poverty, unemployment rates, and housing conditions.
Right. Researchers cross-referenced the Townsend Index with the participants' polygenic risk scores for depression. And they found a profound interaction. Certain specific genetic risk loci only activate, meaning they only contribute to the statistical risk of the person developing clinical depression if the person is also living in an environment with high socioeconomic deprivation.
So if a person has the exact same genetic mutation, the exact same biological hardware, but they live in a highly supportive, affluent environment with low stress, the risk gene essentially stays dormant. It never executes the software code for disease.
Precisely. And the biological mechanism for this is epigenetics. The environment stress, diet, trauma, physically alters the chemical tags on the DNA, specifically through methylation or histone acetylation.
These epigenetic tags tighten or loosen the DNA around the histones, physically preventing or allowing the transcription of those risk genes. The environment physically reaches into the nucleus of the neuron and turns the volume of the genome up or down.
It is the ultimate scientific proof of the old adage. Your DNA is not your destiny. It is a probabilistic map of your vulnerabilities and your resiliencies. The genes build the combination lock, but the environment spins the dial.
And as we look to the future, as we gather more massive multi-omic datasets that combine whole genome sequencing with lifelong data on trauma, substance exposure, and socioeconomic status, we will finally be able to map the precise epigenetic mechanisms of how the outside world alters the expression of our internal code.
Which brings us to the end of our deep dive. We've traveled a massive biological distance today. We started with the illusion of the single broken gene, the Mendel's P's model of deterministic disease.
We realized that was a simplistic fairy tale. Instead, we found a vast, profoundly overlapping tapestry of polygenic probabilities. We looked at how linkage analysis cracked the presenilin scissors in Alzheimer's, how CNVs revealed the synaptic scaffolding of autism, the heartbreak of the false starts in bipolar linkage, and the absolute statistical necessity of 100,000-person mega-studies to find the faint polygenic whispers of depression and anxiety.
And when you step back and look at the entire map, the vision of Medlock Holmes in that chamber, watching the light fracture and branch across the synaptic gap, is more biologically accurate than ever. Psychiatric genetics is the study of a system of immense overlapping complexity.
It is not just about neurotransmitters. It is intimately tied to immune function and microglial pruning, to calcium channel dynamics, to lipid metabolism, and inextricably to the socioeconomic realities and traumas of a person's life.
But I want to leave you with a final provocative thought based on everything we've uncovered. You've seen the data. If familial autism shares its core polygenic architecture with high intelligence and enhanced educational achievement, and if major depression is deeply genetically intertwined with basic human personality traits like neuroticism and profound sensitivity to the environment, are these conditions truly diseases in the traditional sense?
It is a paradigm-shifting question for the entire field of medicine. In cardiology, a calcified, clogged artery is unequivocally a biological defect. It serves no purpose. But in the human brain, are we, in many cases, pathologizing the extreme ends of natural human biological variation?
Is extreme environmental sensitivity the genetic architecture of depression a disease, or is it an ancient evolutionary adaptation that simply becomes burdensome and overwhelming in our hyperstimulating modern environment?
It invites us to completely reconsider the ultimate goal of this science. Perhaps the promise of mapping the human mind isn't to cure a defect or to mechanically fix a broken machine. Perhaps the true goal is to fully understand the unique polygenic hand of cards every single human being is dealt. If we know the exact structural vulnerabilities and the hidden cognitive resiliencies of a specific mind, we can stop trying to force it into a neurotypical box. Instead, we can build an environment, a specialized software where that specific biological hardware can thrive. Think about Medlock Holmes one last time, slowing down the fracturing light at the synapse. He isn't trying to stop the signal. He isn't trying to fix the light. He is just trying to understand all the beautiful, impossibly complex ways it can fracture.